My name is Omar Ford, and I'm the managing editor of MD+DI, and we are at using biomaterial and biomimetics innovations for precision medicine and 3D printing. This is our last panel of the day, last but not least. So I hope everyone has been enjoying the conference so far. It's been a pretty good for myself. But let us begin. Biomaterials play a key role in applications such as tissue engineering and repair, customized implants, drug delivery, and surgical aids. While additive manufacturing enables free-form manufacturing and mass customization, it is also an enabling technology for the clinical implementation of the important precision biomaterials. Our experts will discuss ways that they are using bioprinted materials coupled with precision medicine to tailor treatments that improve patient outcomes. And now I'd like to introduce our panelists from left to right. Sure.
Hello. Hi, I'm Todd Astor. I'm the founder and CEO of Biomembretics. At Biomembretics, we are using microfluidic technology to try to create the next generation of artificial organ devices, addressing the two major impediments to the current devices, which are blood clotting, blood health, and the efficiency at removing gases and solutes to and from the blood. I'm a physician, specialize in lung failure and lung transplantation, and direct the Lung Transplant Center at MGH.
Hi, everyone. My name is Nicole Black. I am the Vice President of Biomaterials and Innovation at Desktop Health. I graduated last year with my PhD from Harvard in Jennifer Lewis's lab, and also was closely collaborating with some surgeons at Mass Eye and Ear. And during this time, we developed a new product that we're calling Phonograft, which is a device for eardrum repair that uses 3D printing and novel materials to mimic the structure of the eardrum and improve placement by surgeons. And so we had spun out a company from that called Beacon Bio, of which I was the CEO and co-founder. We're recently acquired by Desktop Health, which is a healthcare division of Desktop Metal.
Hi, and I'm Jennet Toyjanova. I'm Director of Technical Innovation Advanced Analytics from Ximedica. Ximedica is an end-to-end product development company for med devices. So basically what we do is take a product from idea all the way to manufacturing. We're now part of Veranex, which is a one-stop solution for med device product development, including services like design, engineering, clinical services, market access. I've been with Ximedica for 7 years now, leading early phase development, and I'm excited to be here.
Sure. Awesome. Well, I'd like to ask our first question of the panel. I want to talk about trends in the space of biomaterials and 3D printing of biomaterials. So what's new around the materials, synthetics versus biomaterials, reproducibility, challenges for clinicians and other end users? Jennet, could you tackle that one?
Yes, I'll try.
Yeah, sure.
All right. So, I mean, obviously, there's a lot of development in biomaterials, taking from bioinks all the way to some polymers that you can 3D print with. And the challenges that we're trying to address through all this research and developments is basically helping the end users, the patients. If we need to, for example, and I think Todd can touch up on this, like if we need, for example, transplant, obviously there's lack of, we're behind on how much resources we have for transplants. Can we 3D print them? Can we develop them? So all of this research is kind of taking us to that, hopefully soon, but probably some years from now. So there's definitely a lot of development in materials itself.
Mm-hmm.
Yeah, I can answer that too. Thank you. So one area where I really think 3D printing is going to make an impact is in, using it to impart different mechanical properties into the parts themselves, and using it not just for patient customization, which is what a lot of people think of when they think of 3D printing, but actually using it as a legitimate high resolution manufacturing mechanism. And so our team at Desktop Health, we're really focused on, creating next generation materials that could benefit from the 3D printing and extrusion process, and importantly have benefits for patients through this. And so this is where I think, 3D printing is going to have the biggest impact next, not necessarily with with cell-laden inks and things like that, which are very cool, but very far off from actual commercialization and use, but in more creative uses of synthetic materials and designs through these printing processes.
Yeah, my world really revolves around the care of critically ill patients, and there's probably no more target-rich environment for synthetics and biomimetics than in the intensive care unit. Anything from our extracorporeal devices for patients with organ failure that require filtering of blood, to stents that we use in blood vessels and airways, the vascular connections or cannula that we use in patients, the things that cover wounds, wound care, even down to the containers that hold medications and IV fluids. All of these things that are going to be in contact either directly with the body or substances that go into the body, bring into this whole new area of understanding the interaction with the immune system, blood health, along with the materials that they're in contact with. And this has created just a whole new realm for us to think about as clinicians and as scientists in understanding the materials that we're using, but also understanding coatings and changes that we can make in the surfaces of these materials. And that's really where this whole field of the biomimetics in medicine has come from. So a lot of exciting areas. And in organ transplantation, which is my specialty, the whole future of organ transplantation, which is to put those of us that do this out of work, to make designer organs so that we don't actually need to wait and rely on organs, is the whole future of what we're doing in that field.
Now, how can these current biomaterials evolving, how are current biomaterials evolving to enable the transformation of the design, manufacturing, and delivery ecosystem for clinicians and patients in need of medical implants that enable cellular growth?
Yeah, and so I think it's, again, about understanding the interaction between the body's immune system and the body's blood system with these materials. And so a lot of what we're trying to do is to understand these properties of materials that have these interactions. And so whether it's the material itself, for example, in organ failure devices, thinking about the membranes that we're using, whether it be something like polymethylpentene in our ECMO devices, or changing the surface by adding some of these polymers like zwitterionic polymers to try to inhibit protein attachment, whether it's using biomimetic design, adding synthetic albumin, human albumin, trying to attach anticoagulants to the surface to prevent blood clotting, even using, as you mentioned, cellularization of the surfaces, like using endothelial cells to line membranes to create a much more biomimetic environment for blood substances going through those. All of these are areas that we're exploring and utilizing for our devices in the intensive care setting.
Yeah, I agree with that. Oops, sorry, can you hear me now? That's better. I think two major areas in materials that are going to be developed further, like in the near future. One is biodegradable materials. There's, a small class out there of things like PLA, PLGA, PCL that are used commonly in 3D printing. But these materials don't match the mechanical properties of the tissues in your body and don't quite enable cell adhesion and infiltration as well as other types of biomaterials and natural proteins. And so there definitely is a happy medium where you can design materials that mimic the function of these tissues and the mechanical properties and enable your body to actually harness its own regenerative process during this phase. And so a lot of tissues in your body, for example, we work with the eardrum, are very regenerative on their own. They just really need a scaffold or graft to guide them in this process. And so that's one area where I think a big impact is going to be is developing these resorbable materials that can sort of act as an interim and allow your body to really harness its own natural processes.
I agree with both of you. So I think with all of these materials and their development and also moving closer to having them as an implantable in your body, matching stiffness is definitely key. I mean, there's so much research that has been done where stiffness can change the behavior of the cells themselves, right? So we need to make sure that whatever stiffness of these materials is, it's tunable enough to match your homeostasis, to match the physiology. And actually, like even for product development, what we do at Ximedica, for early development programs, we have to do a lot of testing. And I see a lot of benefits in having these biomaterials matching the physiology is for us to start testing devices at the physiologically relevant environment at the early stages. So let's not run straight to the animal studies. Let's not run straight to the organs. Can we test something and develop something that matches the body environment on the bench? And so that we can start proving the feasibility of the products at the early stages.
There seems to be a tremendous amount of promise for these biomaterials, but I'm wondering what challenges exist and how can the industry begin to address some of those challenges?
Yeah, I think one major challenge that I see is probably regulatory, just finding the right regulatory path for having these products in the body or closer to that. Another challenge that I see, but mostly from the development aspect, one of them is, as I mentioned, is testing. How can we standardize testing of these materials, no matter what they're used for, like doing the proper standardized tests to make sure that we compare apples to apples. Because right now, if you go to every lab has their own test methods. And the other thing that I also see, we see at Ximedica at least, is inspection, right? So we're talking about these 3D printed devices or 3D printed materials that have very unique features on the inside. How can we visualize them on the inside? How can we inspect them to make sure that they are per spec?
Yeah, I agree with that. Also, I think one really important thing is the printer itself and the hardware around that. A lot of people think of 3D printing and they imagine, a MakerBot or an Ultimaker or something like that you could buy, on Amazon or whatever and keep in your house. But there are many different types of 3D printers and 3D printing methods. And, each method is so different from each other. I would also call them basically completely distinct manufacturing methods to begin with. And so these different printers and the materials that you use around them, have different capabilities in terms of both materials and the print resolution. And I think, it's really important for the industry to also innovate in what the ideal 3D printing system would look like for these types of implants and, develop high resolution and reproducible manufacturing, basically tools to enable these new advancements.
Yeah, I think as a physician trying to enter this realm, I think one of the challenges is really trying to identify partners, engineers, scientists, regulatory specialists, really sharing your interests, trying to solve some of the problems you're seeing at the ground level. And I'm not an engineer, and I've come to this, again, through practice and through understanding of what the problem we're trying to solve is, but certainly trying to identify that expertise, particularly in the newer field of bioprinting and 3D printing, and really learning to understand the technology has been a challenge. And then the second would be really, especially with 3D printing involves understanding what you're telling the printer to print. And so really having a good understanding of the biological system that you're trying to mimic is essential. And spatial relationships between cells and extracellular matrix with regards to cellular engineering, even an understanding of how the body interacts with certain materials, that has to be in place before you can actually start to build these synthetic or biomimetic devices. And so those are some of the challenges, at least coming from the clinical side of things.
I want to stay on challenges for a little bit and throw out the F word, the FDA, and talk a little bit about maybe the issues with regulatory challenges or issues that might come up. Has anyone experienced that? Has that been a huge roadblock?
Yeah, so we're going through this right now, and I would say honestly, not with the 3D printing itself. That's one of the lower areas of concern for us. Really, the FDA treats it like any other manufacturing method. So as long as you can show that it's reproducible and you can, you guys hear me? use this, and create, devices that are functionally the same, device to device. That's the same as any other manufacturing method. Really, where I would say the challenge is more so is if you do go down the patient matched route is, fleshing out what exactly the parameters around that. But for actually using it just as a manufacturing mechanism, we've honestly seen more challenges figuring out, biocompatibility testing, extractables and leachables, packaging, sterilization that every other med device company has to go through than we have with the actual 3D printing process.
Sure. I want to segue into something you were mentioning earlier, Todd, and that was about organ transplants, and they're using biomimetic material technology. Can these types of materials have structural elements available for 3D printing solutions for patients that are not possible with traditional manufacturing? And if so, what are the pros and cons that exist for these?
Yeah, so I guess some context for where we are in organ transplantation would be helpful. I've been doing organ transplant now for over 20 years, and we're in this sort of the next stage era of organ transplantation, whereas for the last 2 decades, it's been about trying to figure out ways to just get patients through the surgery, try to understand preventing initial rejection of the organs that occurred right with transplant, to the next era where we were developing new medications to suppress the immune system, realizing that we solved one problem but created another with all the side effects of the medications and certainly the long-term impacts on the transplanted organ. So what we're now looking for is sort of the next generation of transplant, whereas, again, as I mentioned before, we're not needed anymore, that we're able to create organs out of patients' own cells or with designer materials that get around the problem of the immune response to those organs. So that's the goal. The challenge with that is that with our current technology, we can certainly try to mimic certain types of organs in the body that are more simple in structure, like skin, or bone or cartilage or teeth. But when we start to think about more sophisticated organs like kidneys or lungs or livers or hearts, that's where it becomes a bit more challenging, because that's where the spatial relationship of cells and the extracellular matrix and the materials really comes into play. And we don't even have that thorough of an understanding of those relationships in the natural organs to this point. So that's one of the major challenges. One of the other major challenges, at least with regards to 3D printing, is that the ability to create vascularized structures. So all of our organs rely on vascular structures, not only to do their job, but to bring nutrients to the cells within those organs. And 3D printers don't have that capability at this point in time. So although we're very excited about the ability to create or designer organs with 3D printing, it's still, I think, many years off in the future. But where we are today is that we have the opportunity to look at some of the other types of applications, like we mentioned before. And actually, Nicole can talk a lot more about this than I can, about creating synthetic materials and creating biomimetic materials that can use or replace certain types or parts of organs. And actually, we'll have Nicole talk about that a little bit.
Yeah, definitely. Thanks, Todd. So basically, what we're doing is we're developing new materials that are synthetic, but that mimic, the mechanical properties of tissues and also, basically the function of the tissue itself. And so in the case of the eardrum, it has a very complex circular and radial structure to it that's very important to its function. And so if you want to recapitulate this, really the best way to do it is to recapitulate that structure and doing it using materials that are regenerative, so that not only does this device have this structure immediately upon implantation, but as your body is remodeling it, it becomes tissue that actually looks like that structure as well. And so that's one area that we've, really tried to innovate in, is how can we design materials that are synthetic, which is beneficial from a regulatory perspective and from a manufacturing perspective and for shelf stability and sterilization and all those things, but that can actually harness your body's own regenerative potential during that process.
I'm actually excited about 3D printed organs more from testing perspective. I'm kind of repeating myself, but it would be nice to have, if we understand the physiology really well, and yes, putting organs or transplants, 3D printed transplants in a human is definitely pretty far off. But if we understand the anatomy and understand some of the vasculature, it would be nice to 3D print it and put it on the bench and test devices that we are developing. Because again, we're in a world where we're trying to put more sensors in the people, detect things early, detect diseases early. But if we cannot test them, it's pretty much impossible to get anywhere as fast as possible.
Sure. That kind of segues into our next question. How do we get around this? How are we able to test? When do you think that will become available, or what are some of the things that need to happen? What pillars need to be moved so that there can be testing?
Yeah, so I can tell you how we test things right now. So at the early stages of any program, we do a very deep physiological research. So we try to understand anatomy, but not only from the size perspective, but also what pressures are things exposed to, what flows are inside these vessels, how soft it is, so understand the stiffness. And we literally replicate it on the bench. Obviously, we not 3D print the whole organ, but we try to get sub-assemblies from it. Okay, so now we expose this device to these flows because that's what it's going to be in the body. Now we're going to expose it to this pressure because that's what it's going to be in the body. But in order for us to get there, I think there needs to be a little bit more advancement in how we build those structures, because like even if we use bioinks, for example, they're just not structurally there yet for long-term testing. There needs to be some more research and advancements in the material itself.
Sure. Now, I want to ask, can these materials be used for patient matching? And I think we talked about that a little bit, but want to just delve into that a little bit more. Can some of them be used for patient matching?
Yeah, I can talk about that a little bit. And so patient matching, taking the patient's anatomy and working that into your design can be useful in a lot of applications. But one thing you have to realize if you're choosing this pathway is that this means that you basically have an infinite number of designs that you could possibly create. And so thinking of how to validate and test all of these designs is very challenging. And so in our case, when we were thinking about the eardrum, we've worked very closely with two surgeons at Mass Eye and Ear, Drs. Aaron Riemenschneider and Elliot Kozin. And so we've been talking to them a lot about the types of eardrum perforations that they see and the sizes and locations, and really doing a lot of market research, customer research into, do these grafts really have to fit the perforation exactly? Or because since there is some overlap, can we create more of a kit of sizes similar to, what's used in the orthopedics industry for knee implants and hip implants instead that can accompany these? And then that way, if you have a more discrete set of sizes, then it's much easier to validate these. I have seen, patient customization used more so in 3D printing for things like surgical models and surgical simulators. And I think that this is going to be a growing field because for surgeons that are working with patients with very unique anatomies, they can basically create, a model of what it's going to look like before going into the patient. And, since these are not going to be implanted, the regulations around, using them as models are much less strict.
Yeah, no, I completely agree with Nicole. I think that when you look at the technology that we're developing, and we do this work in conjunction with Draper Laboratory, we've talked a lot about the materials we're using and the biomimetic nature of the materials, but a lot has to do with the actual design and the flow of body fluids through those materials as well. So part of what we're doing is creating a biomimetic blood flow path that precisely mimics the blood flow through the human organs, for example, the human lung. And this was done by doing thousands of computer simulations, examining precisely how the blood flows through the blood vessels in the human lung. And then by designing a membrane that mimics that blood flow path so that the physics of the flow, not just the materials of the vessels themselves, are actually what determines the biomimetic nature of the device that we're building.
I would add one more thing. I think when we talk about patient matching, we think implants right away, but there is also like casts, for example. Like there are a lot of opportunities for 3D printing or biomaterials for the external use as well, where we can actually make a lot of headway. And I think in developing countries, it's pretty widely used. Sure.
Now, one of the biggest clashes, or one of the biggest things that you'd have to deal with is moving from academia or a product development incubator. That has its challenges. How do you know when to bring a clinician in to test your product? And how is that relationship usually?
I can jump on this.
Yeah.
So we actually work, since we are a product development company, we work with a lot of startups or a lot of companies that spin out from academia. And our advice, we do a lot of research, early phase development research, where we do reach out to KOLs, to end users, and try to identify the gaps where the design space is. Because oftentimes we have the technology that doesn't catch up with the needs that are out there. So we want to make sure to match those. So our advice always to do it as early as possible in development, because you don't want to be in phase 3 and your validation phase where and have something that physicians say, no, this is not designed for us, or patients say this is also not designed for us.
Yeah, I would agree with that. As soon as possible, the better. I've been working with doctors Ringmeister and Kozin for 6.5 years now, basically when I started my PhD, and they've really been instrumental in actually crafting the project and what we're actually designing. And so coming as an engineer into this project, if I wasn't working with them, I could be, bioprinting cell-laden inks or something like that for this application and not really thinking about surgical feasibility or how they'd be implanted. But the fact that I was working with them so early meant that, I could observe some of these eardrum repair procedures in person and actually see their challenges. And I think not only, having people that you work with closely is important, but looking, outside of your immediate team and, doing customer research on a broad range of populations, because you don't want to, design an entire device that at the end of the day isn't going to be adopted, or say have a device that maybe like 5 to 10% of the market will even be interested in adopting. You really want to figure out what these pain points are and actually have conversations, because you'd be surprised how open and willing clinicians and surgeons are to talk to you about their problems. Like, I feel like frustration with current devices and techniques is one of the top talking points that they like talking to engineers and scientists about. So I think you'd be surprised how willing people are to talk to you and give you their feedback.
Yeah, and I can give the other end of that. I do really like talking to engineers and scientists. And I've been an advisor and consultant for many companies over the years, and it is very common to be brought in much too late in the process. It's thought that they need to have a finished prototype to bring in a physician to really comment on that. And I think the problem with that is that a lot, especially with biomimetics, is that in many ways those first stages have become almost a science project, like to try to create the best representation of the organ or the tissue or the biological system, and not actually setting out to solve a specific problem that the clinicians are facing. And you can bite off too much, than you can chew when you're starting to develop these biomimetic devices. And what you want to be focused on is 1 or 2 problems that a specific material, a specific design, those are the things that can be solved. And you also need to be thinking about the cost of some of these materials and how they'll relate to the cost of the device. Clinicians aren't always in tune to that. Administrators at hospitals are in tune to that. So clinicians may think a biomimetic device looks great and it's something that they would use all the time, but they may not have a grasp of how realistic that might be in terms of the end product. So it's important to bring a lot of different clinical views into the picture, maybe not just the physician.
Sure.
I would add something, Todd. You said something very interesting. I do agree. A lot of companies try to go to the physician when they have a built prototype or something that's very polished. And I don't think it's right, and we don't recommend that at all. Again, that is too late. And one thing that we do is usually we come up with surveys, with sketches, with embodiments. We also build a lot of looks-like prototypes. Perhaps they're not functional, and just talk to physicians about, would you like this feature? Would you like this feature? Because sometimes we could, as engineers, we like sometimes to overcomplicate things. So, and it is not always needed.
Sure. Well, let's quantify that for a second. When is too early and when is too late?
There is no such thing as too early, because for our phase 0 kickoff meetings, one of the first things we do is put together a task analysis. like for all the users, if the clinician is using it, if the surgeon, how does the nurse interact with the product? How does the patient interact with the product? We go through this task analysis in detail and literally like put, okay, here's the gap. This is area of opportunity. Here we can optimize and improve. So there is no such thing as too early. I think getting expert feedback and expert review as early as possible will help the process.
Yeah, I completely agree, but the type of physician you bring in might be different depending on the stage. So at that very early R&D stage, there are certain types of physicians you might bring in that have a really good scientific understanding of the problems that you're trying to solve. And as you get closer to prototype and testing, you want to have some seasoned physicians who understand some of the business side of bringing in these products into clinical practice, and potential physician leaders who understand the adoption of innovation in clinical practice, which is a skill that not all physicians at hospitals have. And so I think it's important not only to identify physicians, but certain types of physicians at different stages of development.
So I definitely shouldn't bring a finished prototype in front of a physician or a clinician. That's enough.
Don't send it out for extractables and leaching testing before you've showed it to a physician.
Sounds great. Sounds great. Well, getting to the market fast is key. So how much is cost a consideration in product development and in the supply chain?
It is a huge, I mean, money is on everything, right, in product development. And a lot of companies that come to us, depending on how big of the company, right, most of the startups have very limited resources. So we need to streamline what we provide to them and kind of help them give as a consulting company, giving them an advice on how to streamline their activities to gain more money or to go back to the investors, to what data to collect. So cost plays a huge role in product development, especially for startups.
Yeah, for sure. And I think considering your timelines too, I think one thing that really helped us accelerate our progress was spinning out from academia and being more of an industry setting, because our goals at the end of the day are less about the science, which you could investigate forever, optimize forever, iterate forever. But once you're in industry, you start having timelines and when you want things to get to market. And honestly, there's competitors out there now. You want to be able to compete with them in a reasonable time frame, and realize when your IP is going to expire, and there's just more considerations than if you were just staying in academia.
Yeah, I mean, a lot of the companies I've advised, it's interesting. They've started out with a slow timeline and using potentially the best materials or the best biocoating that they can use in the system to try to achieve the best results, understanding that those materials may not actually be the ones that they want to use in the end. And to me, that doesn't make a lot of sense from a practical standpoint, because that's going to extend your timeline so far out. And if you're not going to be using that material for the final product, it doesn't make sense to me to sort of to have that moving along and then have to step back and sort of do everything all over again with in vitro testing, animal testing. And I found that mistake making that's been made in a lot of different circumstances. I don't know if you have had that experience of not starting out with the material that you think you're going to end up with. Or...
Yeah, definitely. I mean, working on it every day, every single day, trying to make the best possible thing. But really, our goal is to improve patient lives and bring this device to patients. And just think about whatever procedure it is that you're trying to optimize, whatever condition it is, if there's 1 million of these done every single year, then every day that you wait to put this on the market is like, 1 million divided by 365, people that essentially won't be able to benefit from your device. And, we've been very lucky to talk to a lot of patients that have undergone tympanoplasty with other materials and still have persistent perforations and challenges around that. And so really in our mind is, thinking, is this good enough? And is this going to help the patient significantly more than what's currently available to them? If so, then definitely we would want to accelerate the timeline and bring that device to patients. While in the background, you can also continue R&D on next generation devices around this.
On this topic, a lot of recommendation we have to companies, small or big, is if you want to enter the market with a product that is new and innovative, go with minimal viable product and have a multi-generational plan planned out at the early stages. So a minimum viable product will get to market. You'll get so many learnings from it. And then you already have your multi-generational plan on what the next evolutions will look like that you can learn from.
What happens when that company has to do a 180 though, or has to change gears really fast? What kind of advice would you give them?
Do it.
Do it early.
And do it early.
Yeah, fail early. So if
Yeah.
you have prototypes, if you can do like killer experiments and say this is never going to work sooner than later, I would say, yeah, do those sooner than later.
Sure.
Something is very important to do risk-based approach and identify your highest risk components or features or functions or whatever it is, wherever you think it's a high risk, identify it and concentrate on that, and then everything else will come after.
Do we find a lot of companies failing early and taking that approach or taking the advice well?
Yes, and
Okay.
that's the advice we usually provide, and it actually works because we are a development company, but we have done this for many, many years. So we want to give the best advice possible. So it's in our best interest and in the company's best interest to fail early and to identify those things.
Sure. Now, I would like to ask now, what's one recommendation or piece of advice that you can provide to medical device engineers working in this space? I know we've already said fail early, but what are some other ones that you could probably, just kind of advise or talk about?
My advice would be to collaborate. Yeah, I think there's a lot of teams, there's a lot of companies, there's a lot of research labs in academia that work on very interesting and cool things. And we can definitely learn from each other. And even like in development world where we develop products, we constantly reach out to academia and academic partners to see, hey, what can we adapt? What can we use to be more innovative and faster and solve problems?
Yeah, I think having a diverse team is really important, particularly the mindset of the people on your team and their personalities, which is something that I don't think people think about. In med devices, people tend to be very, very risk averse, which is a good thing, but sometimes that can stifle innovation. And so it's sometimes good to have some people on your team that are more willing to think outside the box and say, well, even though this company did this test, it's not exactly mandated, and we can prove these same things in other methods in more creative ways and actually design better tests around this to do it. And so I think it's really important to have both types of voices in the room to sort of balance each other out and say, this would be the ideal world, this is what we could do versus this is what's realistic.
I think it's pick a singular problem that you're trying to solve within the larger context of the problem that you're trying to solve and do it well. And really take that design to value approach that you're going to create a solution to that one problem, also recognizing the trade-off of costs, and scalability and so forth. So I think the mistake is always just biting off too much, trying to solve too many problems with that one device or that one innovation. And I think that's where companies get into trouble.
So start off small, low-hanging fruit, that sort of thing.
Exactly.
Yeah. Sounds good. Well, now I'd like to open up the floor for questions of our panelists. We have a little bit of time left. So we're going to open up the floor.
I have a question for Nicole about patient matching. And so it seems that there's an opportunity for patient matching in the geometry or the shape of the patient's features. And then there's a opportunity for patient matching in terms of the patient's own cells or the chemistry or the sort of cell therapy of that. And I wonder if these two patient matching problems are actually different. Are they the same? Do you validate them the same way? Can you talk about that at all?
Yeah, definitely. So these are two different, risk categories. It's a great question, by the way. So if you're having, a very standard synthetic material that doesn't have patient cells in it, you can still work with the Center for Devices and Radiologic, Health. But the thing is, if the minute you put, cells or anything in that, you have to deal with another branch of the FDA that deals with biologics, which is a whole other ballgame and set of regulations you have to deal with. And besides that is also the cost aspect. And so, now you're not just thinking of matching this patient's anatomy, but extracting cells from them, growing them in a lab environment, and then putting them into the patient. This is an expensive process, hundreds of thousands of dollars. And so if you're talking about markets where the current standard of care is like, a $500 to $1,000 device, you're never going to be able to make that commercially viable, at least at this moment. And so I would say that's the biggest difference, is which FDA bodies you have to deal with. If it's just devices or if you're putting cells in it, there's a whole other suite of tests that you're going to have to do around that. That's much more complicated on top of this, geometry matching of a synthetic material.
Yeah, and I think from the immunologic sort of answer to your matching, I think that was the second part of your question, was trying to match in terms of matching cells. There's a misnomer that by using a patient's own extracellular matrix or using a patient's own cells to repopulate a device, that gets around all of the immunologic challenges that we face, but it really doesn't, because even a patient's own material or a patient's own cells can elicit an immune response when implanted back in. So there are still a lot of challenges we have to overcome, even with our ability. I mean, this is really all the cellular and tissue engineering that we've been doing for years, but now with the advent of bioprinting has a whole other level to that. And so, so it's the future, but there are a lot of challenges on the scientific level.
I might ask one question for Nicole. Coming out of academia, was it particularly difficult to move into industry? Were the philosophies just so radically different? How did you all deal with that?
Yeah. We were actually just talking about this together right before. Coming out of academia, it's just, you have different milestones. And I think, first of all, the organizational structure is completely different. In academia, usually you have a PI, and then everyone is sort of, on the same level. There's just like a group of, 5 to 100 people that all just report to the PI. Once you move into industry and you start building your team, there is more of a reporting structure, organizational structure, but this makes you much more efficient because you all have a common goal and can really focus within this. So instead of having completely different projects that you're maybe helping with here or there, you have a singular goal. And so I think, this has really helped accelerate us moving out of academia and into industry. And particularly in our situation, we were very fortunate with, the early acquisition into Desktop Metal and Desktop Health, and, providing not just monetary resources, which are very important in med devices. As we all know, it's not cheap to bring a med device to the market, but also benefiting from the expertise of others that have been there and done that, and, have gone through these regulatory processes, I think is a major bonus. In academia, your goal is not to bring something to the market. It's to publish a paper, like, give a cool presentation, something like that. But you're not actually aiming to sell things. And so there's never quite that same push to actually push this through and get it to the market.
I would add to that too. I think it's the way we look at so I didn't go to one company. I work for a consulting company, so we work with different projects all the time. So for me, the huge difference was when I transitioned from academia is that, well, first of all, you don't work on one project day in and day out for 5 years. So that's a big difference. And then the other one is in academia, we work inside out, where you work on technology, work on science, and then the application comes later. Whereas in the industry, in product development, you first identify the problem, you identify the application, and then you go inside to figure out what the technology will be to solve the problem. Because at the end of the day in industry, patient is the one who we're trying to serve. Whereas in academia, it's more about making progress in science.
Mm-hmm. And you also don't have to deal with the F word in academia, being FDA.
You have to deal with PI, though.
Oh, good point. Good point. Nope.
Sometimes you do still have to deal with PIs once you're in industry.
Yeah. Well, that concludes our panel.